Hello and welcome to all of you. Today I will be talking about identification keys. Dear students, as you know, directly comparative methods of plant identification have their own limitations. They are useful only if we already have a rough idea about an unknown plant. Authentic identification of an unknown plant is always done with the help of published taxonomic literature like floras, monographs and revisions. The most essential and useful feature of such literature is the use of identification keys. The keys are by far the most widely used method of identification of organisms and does not require much time, material or experience involved in comparison and recognition. Hence, keys are the most direct way of identifying plants. In biology, an identification key is a printed or computer aided device that aids the identification of biological entities such as plants, animals, fossils, microorganisms and pollen grains. A botanical key is an analytical device for the determination of names of plants. It is a series of specially arranged pairs of contrasting statements. The user is called upon to compare his plant with the first of these pairs, so to decide which of the two contrasting statements applies better to his plant, and then to pass on as directed to the appropriate following pair. This process is then repeated until the plant is named. Now, we highlight some principles of a good key. While constructing a taxonomic key, the guidelines which we are going to discuss should be followed so as to minimize the identification errors, which otherwise could lead to serious consequences of misidentification both in pure and applied disciplines like classification, ecology, medical diagnosis, pest control and conservation, etc. The first is only diagnostic characters should be used at each identification step of a key. The characters taken into consideration should be common to all members of a group and unique to that particular group for which a key is constructed. Number 2 is use macroscopic morphological characters with constant character states when possible. Then avoid characters that can only be seen in the field or on specially prepared specimens. Also, it is important to incorporate as many contrasting characters as possible, but use of geographical distribution characters should be avoided. Now, we discuss types of identification keys. Various types of keys and key formats are used in plant identification. Generally, we group them into two broad types as number 1 is single access keys, also called dichotomous or sequential keys, and number 2 multi access keys are polyclaves and interactive keys. The single access keys work by offering a fixed sequence of identification steps, each with multiple alternatives, the choice of which determine the next step. If each step has only two alternatives, the key is said to be dichotomous, else it is polytomous. Multi-access or interactive keys, on the other hand, allow the user to freely choose identification steps and their order. Now, we discuss these two types one by one. First, single access or dichotomous keys. A dichotomous key is a device for identifying unknown organisms to some taxonomic level, for example, species, genus or family level. These keys, which are the most commonly used in plant identification, were probably first published by J. P. Lamarck in 1778. Dichotomous keys consists of a series of paired statements called couplets that describe some features of the organism. Each statement in a couplet is called a lead. The statements or leads are in direct contrast with each other. The use of the key begin with the first couplet and select the statement that best fits the specimen to be identified. The leads typically begin with broad characteristics and become narrower as more choices are required, ultimately provide the identity of the specimen. For example, couplet 1 has two leads. The first lead of this couplet is seeds round, which signifies soya beans. Then lead second of first couplet is seeds oblong. It indicates that we have to move to couplet number 2. The couplet 2 has two leads with seeds white, which demarcates northern beans, and seeds black, which demarcates black beans. By reading the two statements of each couplet, you progress through the key from typically broad characteristics to narrower characteristics until only a single choice remains. As long as the correct statement of each couplet is chosen and the unknown organism is included in the key, a confident identification is usually achieved. Now, we discuss about constructing a dichotomous key. Taxonomists have different opinions about the design and format of a dichotomous key. Some use numbers to separate the couplets, while others use letters. Some taxonomists place each couplet together, while others may separate couplets. For example, here you see a numerical key with leads of couplet together are parallel to each other, and in example B is an alphabetical key with some leads of a couplet separated or indented as depicted. These two types of dichotomous keys designs which are generally used in plant identification are indented or yoked keys and bracketed or parallel keys. These points become clear when illustrated by means of an actual key. We shall use here a key to some select genera of Ranunculaceae to illustrate the two main designs of a dichotomous key. First, as an indented key, and secondly as a bracketed key. Indented or yoked key. This is one of the most commonly used key in most of the floras and manuals. In this type of key, the subordinate couplets are indented below the primary one at a fixed distance from the margin, the distance increasing with each subordinate couplet. Such arrangement is very useful in small keys, especially those appearing on a single page, but are not feasible when keys run to several pages, where parallel or bracketed keys are more advantageous. An indented key for some genera of Ranunculaceae is shown as the first lead of the first couplet groups plants with flowers actinomorphic. The first lead of the subordinate couplet separates plants having carpels with one ovule, fruit of many achenes, and leaves pinnate. This lead leads to couplet 3 as stem almost leafless, flowers in branched scapes, which refers to anemone, and its second lead is stems with evenly placed leaves, flowers panicled or racemed. This statement leads to couplet 4, which has statements as or leads as leaves opposite woody climbers, which are the characters of clematis, and second lead moves as leaves alternate or whorled perennial herbs. This lead further corresponds to fifth couplet as leaves not radical, perianth calyx persistent, which are the characters of thalectrum, and second lead is leaves alternate or all radical, perianth deciduous. Now, this statement leads to couplet 6. Flowers white or pale, petals 5 to 15, leaves glaucous. These refer to the characteristics of Callianthemum. And second lead is flowers yellow, petals 5, leaves not glaucous, which refers to ranunculus. Now, we come to second lead of couplet 2 again, which groups plants with carpels many ovuled, fruits follicular, or berry leaves by are tripinnate. This is further broken into two leads as flowers, not in racemes, often solitary, large stamens shorter than calyx or corolla, which signifies the characters of Caltha, and the second lead of seventh couplet is flowers small in simple or branching racemes or sometimes in spikes. This takes us to next couplet 8, which has two leads as pistil 1 fruit a berry, which refers to actia, and pistil 3 to 8 fruits follicle, which are the characters of Simsifuga. Now, we discuss second design called bracketed or parallel key. In this type of key, two leads of a couplet are always together and the distance from the margin is always the same. Such arrangement of couplet is useful for longer keys where keys run to several pages. In this type of key, it is easy to locate the alternate lead of the initial couplet and also provides good space economy. Such a type of key is illustrated as first couplet flowers actinomorphic, which leads to couplet 2, and second lead of the first couplet is flowers zygomorphic, which correspond to genus Aconitum. Similarly, second couplet is carpels with one ovule, fruit of many achenes, leaves pinnate, which refers us to couplet 3, and carpels many ovuled, fruit follicular or berry, leaves by are tripinnate, which lead to couplet 7. Couplet 3 with two leads are stem almost leafless, flowers in branched scapes. These signify the characters of anemone. Stem with evenly placed leaves, flowers panicled or racemed, leading to number 4 couplet. Couplet 4 has following two leads: leaves opposite woody climbers, which signify clematis, and leaves alternate or whorled perennial herbs, which takes us to number 5 couplet. Couplet 5 with first lead is leaves not radical, perianth calyx persistent, identifies us to thalectrum. Couplet 6 has two leads as leaves alternate or all radical, perianth deciduous, leads us to number 6 couplet. Sixth couplet starts with flowers white or pale, petals 5 to 15, leaves glaucous, which identifies Callianthemum, and second lead is flowers yellow, petals 5, leaves not glaucous, which identifies ranunculus. Now, we go back to lead second of couplet 2, which is broken into two leads of couplet 7. The first lead is flowers not in racemes, often solitary, large stamens short than calyx or corolla. This identifies Caltha, and the second lead of couplet 7 is flowers small in simple or branching racemes or sometimes in spikes, which leads us to number 8 couplet. Number 8 means that this lead is further divided into couplet 8, which leads is pistil 1 fruit a berry, identifies actia, pistil 3 to 8 fruit follicle, identifies Simsifuga. This is how two types of dichotomous keys are designed. Now, we analyze how to use a dichotomous key in the field. Keys that are developed for regional or national floras often require that plants be collected for laboratory identification, where dissecting microscopes and other necessary equipments are available to magnify extremely small parts which are not discernible with a field lens. Select appropriate keys for the material to be identified. The keys may be in floras, manuals, guides, handbooks, monographs or revisions. If the locality of an unknown plant is known, select a flora, guide or manual treating the plants of that geographical area. If the family or genus is recognized, one may choose to use a monograph or revision. If locality is unknown, then select a general work. However, if material to be identified were cultivated plants, then select one of the manuals treating such plants, since most floras do not include cultivated plants unless naturalized. Read the introductory comments on format details, abbreviations, etc. in a flora or monograph before using the key. Read both the leads of a couplet before making a choice. Even though the first lead may seem to describe the unknown material, the second lead may be even more appropriate. Having compared your plant with the leads and decided which lead fits our plant, we then need to be shown which couplet we must proceed to next. Both these needs are met in keys, however, they differ in different typographical formats as already discussed. In a bracketed key, each couplet has two leads immediately adjacent under the same left hand number. As you see, flowers actinomorphic and flowers zygomorphic, both the leads are side by side. Then carpels with one ovule, fruit of many achenes, leaves pinnate correspond to a lead of second couplet which is immediately adjacent under the same left hand number of the first key. The first couplet to be consulted stands at the head of the key next to the number 1. The next appropriate couplet to consult is indicated by a reference number as 2, 3 or 7 as shown. To further down the key, place it on the right hand side of the chosen lead. These numbers direct the user to the next appropriate couplet to consult. In case of indented keys, each couplet has its two leads indented by the same amount from the left hand edge of the page. The first couplet to be consulted is the one least indented and which has its first lead at the hand of the key. The next appropriate couplet to consult is the one with its first lead immediately below the chosen lead of the previous couplet. Its leads being the next least indented pairs below the latter as depicted. Verify your results by reading a description, comparing the specimen with an illustration or an authentically named herbarium specimen. Now, we see the drawbacks of a dichotomous key. If a key you are attempting to use appears not to work, more likely there may be some deficiencies in the key. A key may fail to work if the specimen you are trying to identify is intermediate in its characters between two of the species that are separated out by the key. The specimen could be an extreme environmental variant or it could be a hybrid between two of the species in the key. Thirdly, it is possible that it may represent a new previously undiscovered species that is not included in the key. These defects generally are likely to be found in keys which are based on study of a very limited amount of herbarium materials, so that the authors was not able to get a proper idea of the range of variations of the species concerned. Another general drawback of such type of keys is that they are area specific. It means that a key designed to identify plants from a particular area may fail to give the correct result if one attempts to use them to name plants from outside that area. Now, we come to next type are multi-access or random access keys. The multi-access key is an identification key which overcomes the problem of more traditional single access keys of requiring a fixed sequence of identification steps. A multi-access key enables the user to freely choose the set of characters that are convenient to evaluate for the item to be identified. The advantage of these keys is that they allow the user to enter the key at any point and the user can choose the most obvious important characters of the specimen to identify. These keys are a relatively new alternative to dichotomous keys and are becoming increasingly popular, especially because of the ease of computerizing them. The multi-axis keys are also called polyclaves, punched card keys, tabular keys or synoptic keys, etc. Identifying organisms with a polyclave is a process of elimination. In a written polyclave, keys there are a series of characters and character states. Each state is followed by a number or code for the species that possess that feature. The user selects any character and then copies down the list of species that possess the feature. Then the user selects another character and eliminates any species not common to both lists. This process continues until the specimen is identified. Such identification methods often make use of punched cards. The two basic types of punched cards are number 1 body punched card and number 2 edge punched cards. Now, first we deal with body punched cards. These cards, also named as window cards or peekaboo cards, make use of cards with appropriate holes in the body of the card. In the process, one card is used for one attribute, that is a character state. Therefore, as many cards are required as number of attributes taken into consideration and the number of taxa to be eliminated are numbered as depicted. Round holes are punched on the taxon number for the taxon which possess the character. The plant to be identified is examined and its characters noted. A good specimen often fetches 20 to 25 characters. One selects those cards corresponding to the characters found on the plant. The cards are put on the top of one another in a stack. Taxa having all observed characters are indicated by holes or windows which are easily seen when the cards are held in front of a light. The logic is simple. Each time a card is added, taxa not perforated in this new card are excluded. With 20 to 25 cards, it is likely that a taxon may be determined or searched narrow to a limited one. Now, we discuss edge punched cards. It differs from body punched cards in having one card for each taxon and holes punched all along the edges of the card, one for each attribute. These holes normally closed along the edge. For each attribute present in the taxon, the hole is clipped out to form an open notch instead of a circular hole along the edge. For identification, all cards are held together as a pack. Therefore, thereafter a needle is inserted in the hole corresponding to the first attribute of the unknown plant. As this needle is lifted up, the taxa containing the attribute would fall down and those lacking that attribute will remain in the pack, lifted by the needle. The latter are put aside and the others are gathered up and the process repeated with them using further characters until only one card falls out of the stack. The taxon which this card represents is identification of the unknown plant. Now, we discuss something about tabular keys. Tabular keys are essentially similar to the polyclaves in the sense that they can take ease of exhaustive list of attributes and are easier to use. The data are incorporated, however, not on cards, but in tables with taxa along the rows and attributes along the columns. The attributes represented in each taxon are pictured with the help of appropriate symbols. The attributes not represented in the taxon show a blank space in the column. Thus, the table will have as many rows as taxa and as many columns as number of attributes for which information is available. Now, we discuss interactive keys. An interactive key is an interactive computer program in which the user enters attributes, that is character state values of the specimen. The program eliminates taxa whose attributes do not match those of the specimen. This process is continued until only one taxon remains. Computer access keys involve the principles of multi-access or polyclave keys rather than sequential or dichotomous keys, and they may not be greatly affected if only one or two of the many characters do not fit. It is relatively easy given the proper computer software to convert multi-access keys to a computer interactive format. Now, we discuss the advantages and disadvantages of these interactive keys. The advantage of multi-access key is that the attribute to be used for identification can be chosen by the user, so that if an unusual attribute is noted, the majority of the taxa are eliminated straightway. Further, these keys are easy to use and faster and easily computerized. The disadvantage is that the cards are relatively cumbersome and time consuming and expensive to construct. Further, only few such keys are currently available and requires a computer be handy when you are making your identifications, which is not problem in a lab, but may be unwidely to use in the field. With this, we concluded our today's topic on identification keys, in which we highlighted various types of keys, how to construct them and how to use them. I hope it will understand you about the structure and functioning of taxonomic keys. Thank you very much.